Welcome you all the delegates for the second session of this day one ASTTP sponsored by AICTE and organized by Hills College of Pharmacy, Tirupati. And we have our today's second session speaker, Dr. Ashutosh Kumar sir. And sir is an assistant professor in Department of Pharmacology and Toxicology. Sir is from NIPA, Kolkata. And to do the honorary introduction, I request my dear colleague, Dr. Jyoti Madam, to introduce the today's speaker to the faculty members. Madam, kindly do the need.
Yes, sir. Thank you, Sucharita. Good morning, respected principal sir, present speaker and delegates. Myself, Dr. Jyoti, Professor, Department of Pharmacology, Seven Hills College of Pharmacy, Tirupati. It's my privilege to introduce Dr. Ashutosh Kumar Garu. Dr. Ashutosh completed his B Pharmacy from Dispar College of Pharmacy, Delhi University, New Delhi in 2004. Master of Sciences and Doctor of Philosophy from Naipur, Mohali, India in 2006 and 2011 respectively. Dr. Kumar professional experience started from 2011 to 12 academic year as scientific writer in Novartis Healthcare Private Limited, Hyderabad. From 2012 to 13, worked as assistant manager in Dr. Reddy's Laboratories, IPDO, Hyderabad. From 2013 to 20 as assistant professor in Naipur, Hyderabad. In 2020, worked as senior resident scientist in Incogen Therapeutics Private Limited, Hyderabad. Present, sir, working as assistant professor in Naipur, Kolkata. Dr. Kumar has international level publications, 56 articles, 2 book chapters, and 10 conference proceedings. And at international level, articles 8, 25 conference proceedings. Totally, sir has 3,350 total citations, 31 high index, and 15.22 highest impact factor. This is from Google Scholar. Dr. Kumar supervised 4 PhDs and 37 Master of Sciences students. Dr. Kumar has 4 membership of professional bodies like International Society of Neurochemistry, Neurodiab, European Association of Studies on Diabetic Neuropathy Study Group, Indian Pharmacological Society, Indian Pharmacy Graduate Association. SAR received many honors and awards like Naipur Fellowship, Naipur Senior Research Fellowship, PC Dandia Gold Medal at annual meeting of IPS, recipient of DST Fast Track Young Scientist. SAR 3 times received International Travel Award from DST and guest editor for International Journal of Endocrinology, Current Neuropharmacology, Oxidative Medicine and Cellular Longevity, and CNS Neuronal Discord drug targets. And also SAR has editorial member for reactive oxygen species. And SAR received Angelica Berhaus Award from Neurodiab and United Reviewer for Strasbourg Institute of Advanced Research, France, for evaluating grant applications and evaluation of research proposal for EMR and Women Scientist Scheme A of DST New Delhi. Dr. Ashutosh Kumar Garu received 8 grants from DST 4, DBT 2, OISH 1, Unicary 1. Majority grants sanctioned amount was more than 25 lakhs. In his career, the best research contributions to science entitled with first one, elucidation of role of NF-kB inflammatory cascade in the pathophysiology of diabetic neuropathy using pharmacological interventions. Number 2, upregulation of AMPK and SIRT pathway promotes neuroprotection against diabetes-induced sensoromotor deficits. Third one, demystifying role of mitochondrial dysfunction in chemotherapy-induced peripheral neuropathy. Fourth one, drug repurposing for neuropathic pain. And fifth one, trauma-induced experimental neuropathy. So thank you one and all for giving this opportunity. Sir, thank you, sir. Over to Sucharita.
Thank you so much, ma'am, for your detailed introduction, actually, and welcome you, sir. Probably it's been a tough moment for me to draft your achievements in a single slide. Actually, it makes me wonder, and also really proud to have you, sir, on this platform of Service College of Pharmacy too.
Thank you.
Welcome you, sir. Thank you for accepting, and continue with the session, sir.
Yeah. So once you stop sharing, then I can start my presentation.
Sure, sir. You can. You can.
Now you can see my screen, right?
Yes, sir.
Yeah, can you see my screen?
Yes, sir. Yes, sir. We can see.
Yeah. So thank you for such a nice and extensive introduction. And at the outset, let me thank the Seven Hills College of Pharmacy and the organizing committee member for choosing such an interesting topic, and which is changing day by day, as you have heard from the first speaker of the session, how the difficulties in changing environment of the research is bringing. So it's a good topic, and I think like participants will, throughout this period of 6 days, they will be learning a lot of exciting things about neuroscience research and all. So in this perspective, I have chosen one topic integrating modern neurosciences tools for studies on diabetic neuropathy. So we start with modern neuroscience, and then I will be discussing about some of the techniques which people have been using recently, and in the end I will be showing some of the results from our laboratory, what we have achieved and all, so that you get a total flavor of like the recent research practices done in the diabetic neuropathy area. So first of all, let me begin with the modern neuroscience. So what do we understand when we hear the word molecular neuroscience? What do we perceive? So something comes into brain, like if we go by the definition, it is the branch of neuroscience that observes various concepts and integrates various concepts of molecular biology applied to nervous system. So this is the basic definition of molecular neuroscience. But it is not such simple thing. It is exhaustive, inclusive areas. Like if you start with molecular neuroanatomy, molecular signaling, cascades, ion channels, nervous systems, their genetics, epigenetics, development, molecular basic of learning and memory, neuroplasticity, degeneration, regeneration, and so many. So it is very difficult to even compile it into a simple definition. So that's why it encompasses all the major areas which are subpart or sub divisions of molecular neurosciences. And we at the present moment has ample technologies advances to understand and to simplify some of them. So going through my presentation, we will be understanding what I am talking in the definition right now. Now, when we take a function of neurons and we tell, like, the basic thing we tell, like, okay, the brain is the most complex organ of the body. Why is it so? It is because it controls the total function of the body. It remembers so many memories. It have lot of chaotic signals, like chaotic traffic jam working in the uh, trillions and of neurons in the brain and the body. So if you see, there is a chaoticness, there is a randomness in the function of brain. But when we see, like, a patient suffering from neurological disorders, Like, for example, if you say a person suffering from epilepsy, in this, the electric signals are very organized. Or if you say, take another example of autism, there is a organized language signalings. That means the multiple random these things uh, are abnormal, normal, and the randomized or the organizing things are normal. So what we say, like in the brain research, that the abnormality, the chaoticness, the randomness in the nerves functioning is new normal, right? So it is very difficult to keep a track of neural complexities and correlate with healthy behavior, keeping track of how the complex processes are working with along with each other, how many feedback mechanisms and cascades are working to enact any single action in our body. For example, if I want to move my hand, there are so many neuronal signals which is working from the brain to the motor neurons and the muscles, right? So that all has to work in a line. Then only the nervous system has to work appropriately. Having said that, you know, the the neurons are one of the highest energy consuming tissues or cell types of our body. Almost 70 to 80% of the cells of the energy, which is derived from the mitochondria, is consumed by neurons. So it is very, very strict protocol of energy generation, energy consumption has to be maintained in the neurons. Any sort of derangement in the energy generation, in the energy utilization, can result in energy depletion, failure of bioenergetic processes in the neuronal cell, can lead to various diseases. And this is where, like, lot of diseases will find their, initiation point, right? And one point I would like to share with all of you, like as I discussed mitochondria, you may be aware that we receive our mitochondria from our mother, right? And mitochondria is only the cellular organelle which has got its own DNA. So it can survive, it can replicate, it can, independently survive in without a cell also. So that is the importance of, mother. Like she is the original energy donator to the whole society. So that is the thing, like we owe our nervous system, we owe our brain complexity, we owe our like brain functioning to our mothers. So it's not to demean the fathers because both has to be in equal ratio to continue the existence of evolution and life. So now let us shift the gears to diabetes because once we have the platform basic understanding of both of the things, then we can go hand in hand. So diabetes, as you all know, is one of the major global challenge which everyone is aware and which is a major threat to the developing as well as developed nation. As per International Federation of Studies on Diabetes, IDF, or American Diabetes Society Association, the overall increase in diabetic is predicted to be around 170% by the year 2035, and it has been renewed now to the year 2045. So almost we will be seeing a lot of impact and lot of increase in the diabetic population in the coming years. And major hurdles in diabetes is there are a lot of complications which are associated with diabetes. They form a major challenge. We have a lot of anti-diabetic medication. We have anti-diabetics, oral anti-diabetics, we have insulin formulation and whatnot, and we can always keep our blood glucose in the normal level. But even apart from this, we are suffering from diabetic complications because they are inevitable. Even though if you keep a very strict control on your blood glucose level, you are bound to suffer from the complication of diabetes, like diabetic neuropathy, diabetic nephropathy, diabetic retinopathy, diabetic cardiomyopathy, diabetic encephalopathy, and so many. So The actual problem in diabetic population is because of its complications later. So that is what we call it. These are the pathies of diabetes, which are making the life of diabetics very susceptible and miserable. Various types of diabetes, I don't think we need to go. Type 1, type 2, we all know. What is the type 3? Type 3 is nothing but Alzheimer's disease, have been also termed as a type 3 diabetes because of its persistent hyperglycemia, mild to moderate hyperglycemia that is present in those patients. Gestational diabetes also, most of us are aware, who are parents, we will be aware, like when a women actually conceives a child, there are chances that during the uh, first, second, or third trimester of pregnancy, she can develop a gestational diabetes, which can resolve once the after the delivery of baby, but in some cases it persists and can lead to lifelong diabetes. Then we have LADA, MODY, double diabetes. MODY is mature onset of diabetes in young patients, and double diabetes is in the patient like having both forms of type 1 and type 2 diabetes. Brittle diabetes is very problematic condition in which the ketoacidosis or the condition of the patient is so brittle that he need frequent hospitalization. And secondary diabetes is due to a lot of medications and a lot of other like pharmacotherapies. One such example which has found relevance in current day scenario is the diabetes caused by the steroids. You may be aware, like in the current COVID situation, people have started indiscriminately using the steroids, right? And but steroids have a major hurdle of, actually, if you use continuous high dose of steroids, it can lead to hyperglycemia, and the blood glucose can shoot as up to 6 folds or 7 folds in the patient. There is, you might have heard like these kind of things in the news channels. People are dying because of overdoses or the side effects of steroids. The second thing is steroids are, immunodepressant or immunosuppressant. So what happened is the patient is under COVID, he is given the steroids, his immunity is going down, and the opportunistic and like microorganisms, fungi, bacteria, viruses are finding an opportunity to invade and infect. So that is where the black fungus, white fungus, yellow fungus, and cytomegalovirus infection can also make the life of COVID patient difficult. So this was different forms of diabetes. Let us try this. I will skip because it is very, very common thing. This is just to show you the impact of the diabetic population. You see, by the year 2040, there will be 642 million patients which will be living with diabetes, and it is very huge. And the condition of India is even worse. Why it is worse, I will tell you. Right. First one is our sedentary lifestyle. Right. And second thing is we are genetically predisposed. Third thing is we have like food habits. Fourth is our sweet tooth is very very strong, and we, for anything we have a sweets. If anything good happens, sweet. Any success, sweet. Any festival, sweet. So everything, like we are forced to eat a lot of hyperglycemic foods who have high caloric value, and that is why our predisposition to get diabetes has increased, and we are ill-fated to become the diabetic capital of the world by the year 2045 or 2050. And very sadly, but I must admit that every third person would be diabetic by the year 2045. So every third person diabetic means in a family of four, you have one or two patients of diabetes, and which is a bigger economic and socio-economic challenges. These are the factors which I was discussing, why we are more prone. Asian phenotype, there are like low adiponectin levels which are present in the Asian and Indian phenotypes. Sedentary lifestyle, we all know we have become, we are in our comfort zone sitting there, not doing any physical activity. Even for switching on the fan, we are using remote now. So that is sort of, lifestyle changes that has come into the present scenario, and which is going to worsen if we don't take, corrective measures. Genotype, diet, definitely diet are also one of the important factors which causes a lot of, problems in the diabetic, in the predisposing the Indian population to diabetes. So, food, I was mentioning, we have a very, for sweet tooth is very strong. We have lot of sweet delicacies. We enjoy eating high cholesterol, high lipid food, high fat containing food, triglyceride food, and all. So that is why these are some of the genes which are there, which are down regulated or not present in the Indian population or Asian population, and which makes us more prone to getting the diabetes. This we can live sedentary lifestyle. These are some of the non-pharmacological treatment which people must use it, because there is a tendency in the new diabetic patient that, like, instead of going for pharmacological treatment, let us make our life healthy. Let us make our diet healthy. We exercise, we do yoga, and we do meditation, so that our body we are training to fight against diabetes. And that is the best solution, I will tell you. Because once you start becoming dependent on the medication, once you give the insulin supplements, there are a lot of burdens which come associated with that. When I will tell you one certain thing, like once you start taking in, insulin formulation, insulin level goes high, then your blood become thick. Once your blood become thick, the chances of getting atherosclerosis, the chances of getting stroke attack, the chances of getting ischemic attack increases. So that is how, like, there is a lot of risk, like, which is associated with most of the drugs which we use. So it's better to go for non-pharmacological therapies to actually improve our body status, to improve our, like, exercises, to improve burning of the calories, to take restricted calories, to make keep ourselves healthy rather than going to the treatment. But it is not advisable for, acute diseases. So in chronic diseases, you still have option to correct your lifestyle, to correct your food habits, and to actually get away from the diseases. Diabetic complication, as I was telling, there are a lot of complications which are associated with diabetes: macrovascular, microvascular. Macrovascular, where big blood vessels are involved; microvascular, on small vessels, capillaries and subcapillaries are involved. So you can see one side we have a stroke, angina, myocardial infarction, cardiac failure, peripheral vascular disease, which are known as macrovascular complications of diabetes. Microvascular complications include neuropathy, nephropathy, erectile dysfunction, autonomic neuropathy, peripheral neuropathy, foot amputation, and all. So these are some of the diabetic complications which the diabetic patients suffer. We will skip this slide, it is more or less repetition. So now coming to the core area of diabetic neuropathy, as I am working for past 17 years in the area of diabetic neuropathy. Since my post-graduation, I have been working in this area. And diabetic neuropathy has, seen lot of translation from the early days in the 2004 to 6, when we used to study only 4 to 5 parameters, and we used to get a good publication. Nowadays, even if you do 40 parameters, it is still not sufficient to publish an article above 3. So you can see the translation. Why I am telling this is because we are going to compare the newer molecular techniques into the current perspective. So lot of changes happen with the advancement of technologies, and lot of understanding, in-depth understanding has increased. So neuropathy, as we all know, it is one of the major complication of diabetes, which has almost prevalence of 40 to 50%. And the symptoms include, numbness, paresthesias, dysthesias. So these are like, once you start developing neuropathy, in the initial period, when your peripherals are sensitized. Sensitized means they are more, susceptible to receiving any response or stimulus. So you will feel a small painful stimuli as major stimuli. So that is called hyperalgesia condition, right? So a small stimuli of a small magnitude is perceived as a stimulus of bigger magnitude. It is amplified. The pain is like felt more. Other thing is allodynia. When a non-painful stimulus like touch or blow of air or touch of paper is also perceived as painful. So in this allodynia, the non-painful stimulus is perceived as painful. There are various forms of single fiber is involved, multiple fibers are involved, small fibers are involved, myelinated, demyelinated aspects and all. Now, so let me take you to the techniques used in evaluating neuropathy. So which in the fag end of this section of my presentation, we will be also seeing some of the modern techniques. So let us start with what are the different models. This is, I think, definitely going to benefit the young faculties or the students who are there in the audience. What are the different models of diabetes? We have streptozotocin, which actually destroys the pancreatic cell, and destruction of pancreas will lead to decrease or rapid decline in the insulin productive cells, and therefore there will be a hyperglycemic or diabetes like condition developing within 48 hours of injection of streptozotocin. Alloxin is also one of the diabetogenic substance which is there, which is used to induce diabetes. But only problem with alloxin is that the intensity of development of diabetes may vary. So some animal may develop very high diabetes, some animal may develop low diabetes. So that variation is very high. So that is why in both STZ and alloxin, people usually prefer the STZ induced diabetes experimental model. Third one is high fat diet induced. We fed the rats with a diet consisting of high lipid content, cholesterol, so that they become obese, they develop the insulin resistance and develop the mild to moderate diabetic complications. Then we have genetically modified animals in which we have genetically modified rats and mice available, which actually develop spontaneous diabetes in their once they become 8 or 10 weeks. So like Jacob fatty rats are there, or obese rats are there. These are certain examples of genetically modified animals available for conducting the diabetes and complication research. Now coming to the various parameters which we do in neuropathy, like we have electrophysiology techniques in which we measure the nerve conduction, we measure the nerve blood flow. We have pain behavioral studies in which we measure the mechanical hyperalgesia, thermal hyperalgesia, mechanical allodynia, and thermal allodynia. Then we have proteomic studies or protein studies, and then we can do genotypic studies or microarray studies where we can study the levels of genes which are affected in this condition. Then we have other imaging techniques used for seeing of what is the state of neurons, how neurons are developing, whether the myelination is intact in those neurons or not. So one by one we will take electrophysiology. Electrophysiology is one of the very important methods in the neuroscience research, and which has started from recording one single nerve cell to the intact in vivo nerve cells. So the action potential, you may all be aware of action potential. That is like there is a change in the sodium and potassium current, there is a change in the conductance, and that is how a signal which is generated from one end, it is carried in forms of various action potentials. So measuring the action potentials in the neuronal cells can give you an idea how the nerves are working, whether they are in appropriate healthy condition, whether they are having some problem, whether they are having some kind of block, whether they are actually demyelinated and all. So this is the thing. Then we have other sensory nerve action potential by which we can understand if the nerves are having axon loss, they suffer from sensory axon loss, and there will be slowing of the conductance. There will be the action potential will be taking longer duration, and due to longer duration, it will be causing a problem in the conducting of the impulses from the nerves. Demyelination, we can also study the demyelination stage, and you can see by uh, assessing the pattern of action potential in the cells, you can understand once the myelin sheet is lost, the conductance of signals will be delayed, or it will be slowed down, and that is what we can actually get here. And we will get a reduced amplitude. You can see here there is a reduced amplitude of the action potential. So that means the nerve cells have lost some percentage of myelin sheeting, which is causing the slowing of the neurons. And this is how we measure the nerve conduction velocity in rats. We can use the tail for recording the sensory and motor nerve conduction velocity, or we can use the tail one, sir, and we can use the paw of the animals. So what we do in this studies is we actually stimulate at one portion of the nerves, and we receive at the other portion of the nerve. And the same sort of studies are done for patients also, when we actually stimulate here in the hand and we receive it here. So that means we know the distance, we know the how much time it took from the signal to start from here and reach to here, how much time it has taken. So we can calculate the velocity. We have distance, we have time, so we can calculate the speed and velocity. So that is how the motor nerve conduction velocity is is measured in the rodents. We also do using the Power Lab ADI instrumentation that we have with indigenously made electrode system that we have only made at our lab. The second experiment what we do is like how do we measure the nerve blood flow. The earlier speaker was mentioning about the ischemic blood flow measurement, right? So here we also use the equipment to measure the blood flow of the nerves, whether it is sciatic nerve, whether it is skin blood flow, or even we can do a drill in the skull of the animals, and we can also measure the cerebral blood. So what will happen in case of a stroke or in case of ischemic attack, the blood flow to that area will be abrupted or stopped. So you can see there will be a decrease, sharp decline in the blood flow in the cranial nerves. Similarly, in diabetic condition, the sciatic nerve, which is one of the most prominent nerves supplying to the peripheral limb and originating from the L3, L4, L5 of the vertebral column of the animal. So it will be affected. So we expose, you can see here, it has been exposed using surgery, Meider surgery, and then we will use a laser Doppler probe, which has one probe to give a light and other probe to get the reflected light back. So whatever the blood flow in the nerve will be there, it can be arbitrarily converted by the software to the blood flow perfusion unit, and that is how you measure the blood flow. So these two techniques are used to measure the functional parameters of the nerves. And there are so many other techniques like in brain things. You can actually record the various signals in the brain, and you can actually conduct the diagnosis of various tests. Then coming to the behavioral testing, I think most of you will be aware, like we do a lot of testing to study the pain, how they are perceiving the pain. So we can use hot plate where we will be heating the plate at some specific temperature level as per experimental requirement. Like for our experimentation, we used to keep it at 45 degrees Celsius. And we'll find out like how much time the animal is able to overstand that temperature, and or doing some kind of giving some kind of the signals that it is feeling the stress of or pain due to the heat. It will start biting, it will start jumping from the hot plate. So these all are like the signals of struggle, as considered as the signals for this thermal hyperplasia. The other parameter what we use is using infrared light, in which we house the animals here, and then we have a infrared source here from this down, and it will be incident on the paw of the animal. So the moment it feels the heat, it will remove the paw. So once it removes the paw, the current circuit will be broken, and then the reading will be, the time point will be recorded. So it will also record and help us to understand how much time or threshold for sensing that pain stimulus is required by the animals. Then we have one-way anesthesiometer, one-way fibers, which are used to study the mechanical hyperalgesia and mechanical allodynia in the animal. And you can see with this graph the animals which are feeling, which are controlled, they are having a high level of weight threshold. That means they are sensing that threshold at higher, weights. So these higher weights means they are normal. But in the animals which are having like a diabetic or something like that, they will be having a lower, threshold. So they will be taking lesser time to feel the pain because of their nerves sensitization. Now, coming to the anatomical studies that most of the people do, one is the nerve morphometry. Morphometry means a term tells about like you are measuring the structure or morphological features of the tissue. So in nerves, the morphological structures would be the myelination and demyelination and number of large and small fibers. So what we do, we take a portion of the tissue as a skin biopsy, and we actually take a skin biopsy, and we take a specific thin section of the skin and then do the histological studies. So we measure the intraepidermal nerve fiber which is present in that section of the skin. So usually there is a lot of A fibers, A delta fibers, and C fibers which are present in that section, in the thick skin section, and you can actually you can see these are the skin section of the animals. We have collected it from the paw, and this skin we have cut into and fixed with that, and we have actually um, stained it with PGP 9.5. But it is a neuronal marker. So you can see the nerves, fibers which are appearing in a control animal, and very less number of nerves you can see in the diabetic animals. And again, like with some treatment, our aim is to replenish the lost nerves in the sections. So you can understand, you can means clearly see the difference between the control as well as the diabetic animals in the skin biopsy or intraepidermal fiber. And it is one of the gold standard techniques which has been used. This is using a fluorescent tagging, and you can see here also the PGP 9.5 antibodies have been tagged, and this fluorescence nerve fibers, you can see a lot of them, and can quantify like what is the extent of nerve loss seen in the patients is compared to normal condition. Now, one more interesting thing I would like to discuss here with all of you is corneal confocal microscopy. It is one of the newest technologies which has been developed by one of my collaborators at University of Manchester, and their group is extensively using this technology to study the various aspects of nerve degeneration. So I will be telling more in more detail. So this microscopy uses a confocal microscopy, which actually put a light source in front of your cornea and illuminate the cornea, and then like a normal microscopy, it takes the picture of corneal layer. So why cornea is chosen? So there is a region, cornea is one of the most densely populated or innervated tissue in our body. It has almost like 6,000 nerve fibers per square micrometer, which can be seen in the cornea. And any kind of neurological diseases or neuropathological condition which are occurring in the body are definitely going to be reflected in the cornea, right? And this equipment is very simple. How you go and check for your glasses, the number, what is the number, what is the lens, whether your vision is correct or not. So same like that, optometric microscopy we do. And you can see the patient will be set to look into the microscope, and within 2 minutes, the cornea will be scanned. What are advantage of this system is, number one, it is non-invasive. Just like eye checkup, you go and walk into a clinic, take 20 minutes with 2 minutes of scanning, and you are done, right? And the results outcome are very beautiful, and it is well correlated with skin biopsies. So the skin biopsies, which were considered earlier as a gold standard for studying the nerve fiber density in the skin, now this technique completely correlate with that, and the results are very much comparable. So that is why, and but still it is in very nascent stage and very less labs, and I don't see any lab in India, maybe one or two facilities like LV Prasad Eye Institute in Hyderabad or AIMS, they may have this facility, and which need to be further developed. Now I will show you, this is the corneal microscopy picture of my cornea, because when I visited my uh, collaborator in the Manchester. So they have a clinic where they used to receive a lot of diabetic patients. They used to study the post-bariatric surgery and then after surgery, and then they want to see like what is the kind of changes that is appearing in cornea. They also, study many kind of, patients suffering from neuropathic condition already. So you can see here in the first column, you can see the starting from 50 this 0 micrometer from the epithelia of the cornea, the slowly till 50 microliter you will get the epithelium cells. At 62 micrometer, your nerve fibers, you can see this, nerve fibers will actually, I will just take a pointer here. Yeah. So you can see here that these nerve fibers are visible at 62 to 70 micrometer. Within 10 micrometer, you will get these all nerves. And once you cross, you come to 70, 80, 100, you will see the stromal cells, posterior stromal cells, and then the other side of the epithelial cells. So this is how it is very beautiful, required no staining, only non-invasive 20 minutes eye checkup like requirements, and you can evaluate how many nerves are intact in the cornea. That means if there will be any neurodegenerative disease which is there, or in diabetic, the numbers will be less. So this is healthy. As I told you, it is my cornea, and we will see like how we will evaluate. So what they will do with the help of software, they will trace these main neurons, they will trace the branching, and they will calculate various parameters like nerve fiber density, nerve branching density, and other parameters. So this help us to evaluate the number of branchings, the number of intact nerve fibers bundles which are present. In diabetic condition, what will happen? These main branches will be reduced. So in this frame, you can see almost like 5 to 6 main nerve fibers which are traced in the red color, right? But in diabetic, you will hardly see one or two, and that too with very minimal branching, because for nerve functioning, the branching is very important. The more they branch, the more healthier the nervous system, the more healthy is its functioning. So in diabetic, it will be decreased. So very good technique, very easy tool of neuroscience, and very much relevant for neurodegenerative disorders, even Huntington, Parkinson, Alzheimer's, psychiatric disorders, epilepsy, wherever nerve fibers are deranged, and can be definitely studied using this, and we can get a hint, okay, what sort of nerve health is present in this fellow. Next is patch clamp technique. You all be knowing like patch clamp is one of the very sophisticated techniques which is used to study the single cell, single cell nerve ion activity. So in a cell where there is a lot of ion channels are also there, like receptors, sodium channels are there, potassium channels are there, which actually radiate, which actually pass on sodium outside or inside. So these voltage gated channels will help us to study like how the sodium current is working, potassium current is working. And this is patch the single cell with the help of microscope with the that of a trained for doing during my PhD. So what we do, you can see this is a cell neuron, this is a electrode pipette, and it is you know patched with the cell. Now you can see a ion channel represented here in green. So it can actually study like what is the movement of ions or what is the movement of sodium through that channel, and it can compare the sodium current with that of healthy. How in diabetic nerves or the nerves which are cultured and or collected from the diabetic patients, how the sodium current is working. So this gives an very interesting results and very translational impact of this parameter is very high because directly it gives an idea how your nerves are working, what is the health of your nerves in terms of iron functioning, in terms of sodium current, because sodium, potassium, lead action potentials are the basis of conductance of the neurons. These are some derived parameters which I think we can skip, but the only understanding you have to do is like we can measure the currents, we can measure sodium and potassium current, which will tell you how in a cell these voltage gated channels are ion channels are working, and that will positively correlate with nerve function. If the current is working fine, if the current is good, then you can see the nerve functioning in normal. Other one is calcium imaging. So this is not from our laboratory. I have taken it from like the expert article published by some of the researchers in USA. So they will be using a calcium fluorescent based calcium imaging, because in the neuronal cells, in the neurons, the calcium is playing a very important role because it directly relates to the release of neurotransmitters. It directly relates to the how much a neurotransmitter should be released so that the action or the conductance will be carried out. In case of the diseases like epilepsy, there will be high levels of calcium which is going on, and due to which there will be hyperexcitability in the neurons. There will be excessive firing of the neurotransmitter at the junctions, synaptic junctions of the neurons, and there will be a lot of neurotransmitter available, which will lead to overactivation and excessive neuronal activity in those subjects. So this is one of the good techniques which can be used. Other one is silver staining. Silver staining is one such technique which has been used since many decades, and it is used to see like what is the extent of nerve regeneration or degeneration. So in this pictures you can see the cells which are uh, stained with black, so they are dead neurons. So the level of dead neurons can be studied, and you can study in cell like in disease condition or in any mimic like experimental condition. Like for example, diabetes, you expose the cells to hyperglycemic condition, and you find out how much of the neurons after 48 hours of hyperglycemic exposure degenerate. So that also as an important way of studying the impact of the various treatment in the neuronal cells. Now, I told you in the beginning that mitochondria concepts, and it is very important. Mitochondria, why mitochondria is important is, I told you 70% of the body's energy generated or ATP generated in the body by the mitochondria is consumed by the neurons. So it is prerequisite that the neurons which are there, they should have high mitochondrial content. They should have like high healthy mitochondrial content. Excuse me. So we need healthy mitochondria to have a healthy neural system, to have a healthy nervous system, and to actually, have a proper functioning of the nerve cells. So that is why when I started my research as a faculty at NIPER Hyderabad, we initiated the studying the mitochondrial dysfunction in the neurons in with respect to neuropathic pain. So one by one, let me take you through that. So mitochondria, we all know, is very much important for neurons. And mitochondria during its phosphorylation, complex activities 1, 2, 3, 4, 5, it generates ROS, right? And there is a mechanism to neutralize that ROS. But in the conditions where there is a disease-like condition, what will happen? The capacity to neutralize the reactive oxygen species which are formed in the body is decreased. The antioxidant defense system, which is present, the proteins related to antioxidant defense system, they decrease. And due to this decline, what will happen? That the level of free radicals will increase into the mitochondria, into the cell, and then it will lead to the condition called oxidative stress. So we all know, like most of the researchers, I am sure everybody has exposed to like the scientific literature related to oxidative stress, because it is the basis of most of the diseases, I can say. So this is the region. Mitochondrial activity directly contributes to oxidative stress if there is no proper functioning of mitochondria. So one more small story I would like to bring here. Like in early 70s, 80s, we used, or 90s also, we used lot of antioxidants. We studied lot of antioxidants, right? We have shown very good results in animal studies, but those antioxidants, they failed when they went to the clinical trials. Why they failed? Why the translation of our animal study was not possible when it went to the clinics? Even like lot of stroke research, which was established in the early 2000, you can say, or 2010, you can say, most of the stroke research did not see the light of clinics. Why? Because the translatability is less, because we were targeting the radicals which are formed and neutralizing with antioxidant. But nobody thought of, why not target the source of free radicals? Why not target the mitochondria? Why not keep the mitochondria healthy? So once the mitochondria is healthy, it automatically produces less superoxide, and whatever superoxide it produces also, it neutralizes it. So it become evident that mitochondrial functionality is very critical, and we started working onto it. So now how we started, I will tell you. Like first thing is, we started to target the proteins which are related to mitochondrial functioning. So we started with AMPK. What is AMPK? AMPK is adenosine monophosphate activated protein kinases, which works as energy sensor. Like when there is less energy in the body, AMPK gets activated. It gets activated, promotes the mitochondrial functioning by either mitochondriogenesis, right? Or even it can lead to activation of pathways leading to mitochondrial fission or fusion as the pathological condition is required. So it will lead to improved energy status, and again, like through metabolism also, it maintains the basal level, right? But in diabetic condition, the AMPK was decreased. The AMPK levels decrease means there will be a problem in maintaining the energy homeostasis. And in diabetic condition, we have excess of glucose, but glucose is not being able to be utilized. Therefore, it remains in the systemic flow and damages most of the tissues and organs. So we activated AMPK using synthetic molecules, natural molecules, and we found that AMPK modulation has actually improved mitochondrial functioning and also have given good results in terms of nerve functioning and protecting against neurodegeneration by the diabetes. Then again, in the second phase of our studies, we tried to link how AMPK activation, because per se AMPK directly cannot impact the mitochondria. So what are the in-between signaling cascades? So we have started from one end of the thread. Slowly we reached to SERT1, SERT3, NRF2, PGC1 alpha. So these are different proteins, transcription factors, which kinases, which are related to the AMPK downstream signaling, linking it to the level of mitochondria, right? So let us see. This I think we will read. So what we did, we have started using the animal models, as we use the streptozotocin induced animal model and to induce diabetes. And if we keep the diabetic animals for 6 weeks, it will develop neuropathy. So we will give treatment in 7th to 8th week, and then after 8 weeks, we will sacrifice the animal. Sometimes treatment is 2 weeks, sometimes it is 4 weeks, sometimes it can be from the starting when we study the preventive regimen. So this was the experimental design for elucidating the AMPK activity, and neuro-2A cells were used, or PC12 cells were used, and they were made differentiated into neuronal cells and then exposed to high glucose and drug, and we found out what kind of changes are happening. So let us see. First is the this study we did with one of the synthetic molecule, bordoxylin methylate, which is one of the NRF2 activator, and which is there in clinical trial for diabetic kidney-like conditions. So diabetic kidney disease, it is there in clinical trial recently. But we started this work in the year, I think 2017, we started this work and published this whole studies by the 2020. So in that we found that the diabetic animals, you can see, this is nerve conduction velocity, what I was telling in the electrophysiology slides. So you can see the control animal, which are having a nerve conduction velocity of around 65 to 70 meter per second, was decreased to around 35 to 40 meter per second. And our treatment improved it. Similarly, the sensory nerve conduction was also improved. Nerve blood flow was also improved. That means our treatment, this bordoxylin methylate, which is an NRF2 modulator, it improves the nerve function. And why it improved the nerve function, that we will know in the next few slides. This is the intraepidermal nerve fiber density with PGP 9.5. You can see the green fluorescent seen here. They are the nerve fibers, right? So this is the control animal, and this is a diabetic animal. You can see, you can count number of this cells, around 5 to 6 are visible, but here it is only 1 or 2. So that means the epidermal nerve fiber density was decreased in the diabetic nerve skin. You can see the numbers of treatment has increased the numbers to at least more than double at that of diabetes. So it also improved the nerve fiber density. Now coming to the mechanism, why it given the neuroprotective effect. So bordoxylin methylate is an NRF2 modulator. NRF2 is a transcription factor which is involved in maintaining the antioxidant defense. So once NRF2 transcription happen, it leads to the production of proteins like HO1 and QO1. These are the enzymes which are linked to neutralizing the free radicals generated. So this is like improving the antioxidant like defense in the body. So you can see the NRF2 label where we have done the NRF2 immunohistochemistry in dorsal root ganglion. Dorsal root ganglion is a connection of nerves from the L3, L4, L5 of the vertebral column or spinal cord region, and it connect to the spinal sciatic nerve. So this is very important tissue which you hardly get like 5 to 10 milligram per animal. You will get the DRG tissues because it is a hardly comprised of around 100-150 neurons. So it is very small tissue which we collected. We have done the immunohistochemistry, and you can see the cells which are here stained at brown. They are the diaminobenzidine stained NRF2 positive cells, right? So you can see there is a lot of numbers are there in the treatment, but in the diabetic you can see very less or almost negligible, right? So this is to prove that, okay, this drug molecule is improving NRF2. So once it improves NRF2, automatically it will improve the antioxidant defense, and SOD being superoxide dismutase being one of the neutralizing enzymes present in the cells. So you can see the SOD labels with improved NRF2 labels, SOD labels also improved. That means with NRF2 upregulation, SOD upregulation also was there. That means SOD, more number of SOD is present in the treatments. That means it is able to neutralize more number of free radicals which are generated. And that is how we establish that, okay, it is also working in the diabetic animals where it is protecting against the oxidative stress. Then again, to confirm these all results, we did the Western blotting. You can see NRF2, KEAP1. NRF2 and KEAP1 are coexisting molecules. So till KEAP1 is attached to NRF2, it keeps it in inactive position, state. So for activity, NRF2 must get rid of KEAP1. So you can see the KEAP1 in NRF2 in the diabetic hyperglycemic group or diabetic group, very less number is very less intensity of band you can see. That means the levels of expression of NRF2 was decreased in the diabetic animals, and with treatment you can see very much improvement in the NRF2 level. SOD level also improved, NQ1 or HA1, which I was telling, that antioxidant detoxifying enzymes also increased, and other proteins. So this I will not go into that these details, but this heat shock proteins levels were also improved in the treatment. And heat shock protein is one aspect of protein quality maintenance in the mitochondria. These are stress related proteins which are reduced in the diabetic condition, and you can see all of the heat shock proteins have decreased here, and in treatment it increased. So they maintains the protein repair process in the cells, in the mitochondria. That is how our treatment improve their levels, means our treatment also causes improvement of protein quality control in the mitochondria, contributing to keep it healthy. Now, the one more technique we use is co-immunoprecipitation, in which we will take or select two proteins, so lon P1 and SERT3. So what is lon P1? Lon P1 is one of the ATP dependent proteases, which is involved in repairing process. So in mitochondria, when any kind of proteins are damaged, so first they have to uncouple them, they have to make them structurally damaged proteins, and then use these components to make the newer cellular component. For example, if there is a dysfunctional protein is there, so it will digest with the help of like proteases with the help of autophagic processes in the mitochondria, which is known as mitophagy. And after digestion, whatever the important parts which are available now will be used to regenerate or make newer, healthier components of mitochondria. So that is how the structurally damaged proteins are taken care by lon P1 and heat shock proteins to keep the uh, unhealthy proteins or damaged proteins, either they are recycled back to make the healthier component, thereby improving the health of the mitochondria and also preserving the active mitochondria. So this is how like lon P1 is related to mitochondrial dysfunction. In diabetic, we have proven that lon P1 levels were decreased, and then why there was a need to do immunoprecipitation is because these two proteins, SIRT3 protein, SIRTwin3 is present in mitochondrial membrane. Lon P1 is ATP proteases responsible for mitochondrial protein repair process. So whether these two coexist, that means if one is there, other labels are increasing or not. So that we have co-immunoprecipitated, and we were able to prove that lon P1 and SIRT3 go hand in hand. So if there is a treatment with bardoxolone, it has improved both lon P1 and SIRT3. That means they are interacting with each other and causing the repair of the proteins or damaged proteins in the nerves which are exposed to diabetic condition. So this study helped us to understand, okay, in diabetic condition, lon P1 and SIRT3 levels are decreased, and our treatment with bardoxolone methylate has been able to improve the lon P1 activity as well as SIRT3 activity. Complex activities we have done in the mitochondrial fraction and of the like DRG collected from the diabetic and the treatment animal, and all the complex activities you can see were decreased in the diabetic animals, right? So that means mitochondria are not healthy, they are not properly functioning. The phosphorylation oxidation in the through the complex activities of the mitochondria is compromised in diabetics. And treatment by improving the mitochondrial activity, this may be also they are improving the complex activities, thereby making the mitochondria to work in a better way. Again, these are cell culture studies. I think similar proteins we studied in the cell culture model also. So this slide we will scope. These are confocal microscope images. You can see NRF2 proteins in the diabetic, which is represented at green fluorescent, was decreased, and the treatment has replenished this green fluorescence. That means it has replenished the NRF2 levels in the treated neuro 2 cells. So it means that it has helped us to not only fight the reactive oxygen species by influencing positively the NRF2 modulation, also it is helping to cause the mitochondrial protein repair using this kind of proteases. So again, some studies, further studies for generation of free radicals using DCFDA, and we have done like DCFDA and with the absorption studies, and also mitosox, so mitochondrial superoxide levels. So this experiment has suggested that in hyperglycemia there was a change in intensity. That means there was a more number of DCFDA intensity correlate more number of free radicals which are generated. So this was there, and then we have also conducted the flow cytometry assay for mitochondrial membrane potential, because mitochondrial membrane consists of so many of the important regulating proteins like cytochrome C, apoptotic inducing factor, and so these factors are responsible for controlling the processes like apoptosis, and also like formation of inflammasome, which relate to the inflammation later. So what will happen in diabetic condition, the nerve mitochondrial membrane potential will be changed. So therefore we will see a lot of red to green fluorescent changes in the hyperglycemic group, and treatment group almost replenished it back like that of control. So treatment also improved this and prevented the mitochondrial membrane potential changes or mitochondrial dysfunction. Again, this is one of the very exciting experiment that nowadays people are doing. It is a seahorse equipment, and seahorse is used to study the mitochondrial functioning into the in the isolated media. So what we will do, we will culture the cells, and in that cells we will actually measure the oxygen consumption rate, the extracellular acidification rate, or the amount of like proton generation. So we will give certain inhibitors like complex 1, complex 2, complex 4. So these are the like oligomycin, FCCP, rotenone, or actimycin A. So these are inhibitor of specific complex. That means there will be a change in the oxygen consumption rate once we block complex 1, once we block complex 4, complex 5, like that. So this is plotted, and with this plot we can actually come to know what is the oxygen consumption rate of these neurons which are cultured here. So you can see the oxygen consumption rate is decreased. That means the metabolic processes are decreased, the mitochondrial function is decreased. So in the hyperglycemic group. So this also again help us to understand that there was a compromised mitochondrial functioning in the diabetic nerves, and which was replenished back by our the drug here. So summarizing the overall effect of this study, what we have done. So you can see there is when there is a glucose hyperglycemic condition, it will cause the changes and alteration of complex activities. There will be more ROS which is generated. ROS will damage the proteins, and it will be misfolded proteins will happen. If heat shock proteins are there, they will make them folded and correct them. If they are not there, then these proteins will be undergoing the degradation process with ln P1, and with autophagy or mitophagy, it will be recycled back, right? And you can see a lot of pathways related to STT3, and even we have like something AMPK role here. So AMPK modulates the NRF2, but here we have used the bordoxylin, which is actually activating NRF2. Activation of NRF2 will lead to the transcription of proteins like NQ1, HO1, SOD2, STT3, PGC1 alpha. PGC1 alpha will be useful for generation of more mitochondria, or it will promote the mitochondriogenesis. These three will be helping to strengthen the antioxidant defense. So whatever problems which are coming here, like whether it is ROS formation, it will be neutralized by these. Whether it is misfolded proteins by ln P1 and HSP, it will be made into folded, or if not folded, they will be degraded and recycled by autophagy processes. So like that, we identify by utilizing lot of these molecular techniques what will be the probability mechanism, and each mechanism was proved by using one or two actually different modalities to come to the same conclusion. Because in research, it is very important to prove the same concept using 2 to 3 different ways, so that whatever you are claiming, it can be proven from 2 to different perspective angles. Then it can be actually your claim can be verified, and you can take it forward. So that was the total story which I just shared you with one portion of our studies, where we have tried to use these various molecular neuroscience tools, and to actually derive the conclusion that yes, AMPK or NRF2 modulation can actually add the mitochondrial functioning in the neurons, which will help us to keep the neurons healthy, functioning, and protect against various neurodegenerative disorders. So what are the take-home lessons from this session is, like, we have with the advent of technologies to make the cutting edge research, right? But again, to understand, the more we understand, the more complexity we will discover. So that is where we have to sometimes, take the responsibilities and use this knowledge in an appropriate manner to make some sense and to make like some of the discoveries which can actually help benefit our society. We know, like, with more power comes a bigger responsibility. So with all these things, with all these technologies, we have to use it ethically to be to better the humankind, with many wonderful things unfurling in the last decades, like artificial intelligence, machine learning, Internet of Things. We can really take these all things to demystify the complex nature of diseases and discover better therapeutics in the coming days. So that, like, we contribute to humanity, we give back to the society, and that is what the mantra, contributing to our well-known ideology of Vasudhaiva Kutumbakam. Everybody should be healthy. Everybody should, come over the sufferings and pain. So that is what we also believe. These are some of the publications which have come out of my group. And this is my research group with the beautiful team here. And this, whatever work I have presented here, whatever publication I have done, it is all work done of my students. I was the guiding force for them, providing the parently support to them, but all the work credit goes to all my students. This is my new team at NIPER Kolkata, and we are developing this group here also. And very soon we'll be able to re-establish some of the newer techniques and will start contributing further. I will, nevertheless, I would also like to acknowledge a lot of funding agencies which have given funding to support my research, because the more complex parameters you add into your research, it will implicate more cost to it. Like once I showed you the seahorse experiment, one seahorse experiment cost us around 1.5 lakhs per plate. So that is how, like, we will be needing a lot of fundings. And I was very lucky to be receiving the funding from the STTP, UKRA, and I have international collaborations also with Manchester and in Poland laboratories. Thankful to NIPER Hyderabad, NIPER Mohali. Mohali I was trained. NIPER Hyderabad has given me opportunity to work and establish my work and do all these wonderful things. NIPER Kolkata, where I am presently working. These are some of my collaborators. So thank you all for your patience listening. And I hope, like, all of you stay safe, get vaccinated, and keep yourself healthy, because future is ours. We will beat Corona very soon, and we will be again coming back to the normal things. Thank you. I would be happy to take on a few questions if time permits. Thank you.
Yes, sir, definitely. Thank you for your wonderful deliberations, actually. And you clearly emphasize the importance of biochemical pathways in alleviating a few disorders, especially the diabetes, neuropathy.
Yeah.
So it was very nicely presented, sir. And kudos to your team also, because your success has been self-replicated in the work of the teammates.
Yeah.
So with your permission, we have a few more questions in the line. Can I take up, sir?
Yeah, definitely. Please go. Right.
Yes, sir. So here's the question from Dr. Atanu Bhattacharya, sir. So he's asking a question like, how to estimate the baseline in rat brain homogeneity? So you can check the question chat box.
Yeah, I can see it. So actually, our relation means our studies are mostly to the peripheral, peripheral nervous system. But you can actually, study this base one by using some of the, I think, the PCR techniques or the microarray techniques, which will help you to understand, like, how base one or amyloid things are related to, specific regions. So you have to understand what of the specific brain region to be, we have to isolate and to develop the for your this microarray or proteomic analysis. Right. You can find many procedures. I'm not sure, and I'm not the correct person to comment on the procedure, because we don't do it here. Right. But you can get a lot of data on base 1 and APP regulations in the brain samples. Okay.
Yes, you can see a lot of positive messages in the chat box too, sir.
Yeah, I don't see any further questions, I think.
Yeah, questions. And requesting any of the faculty members, if you have any query, kindly unmute your mic and pose a question so that we can make use of this opportunity at the best. So any faculty member, do you have any query? Yeah, I hope there is no queries from the participants.
Yeah. We are very close to lunch session. Maybe that may be a reason their glycemic levels will be down. So everyone is eager to go for lunch. So yeah, fine. Anyway, I have you have my mail IDs. If any kind of questions you develop or you want to ask or clarify, it will be, I will be happy to do that.
Yes, sir, definitely. If the participants need, definitely we'll be ready to share the contact details and mail ID of yours too. So kindly do the support to all the faculty members.
Yeah.
And so to do the honors, we are much gratitude. We have to express so much gratitude to you for accepting the invite, actually, sir. So even for you too, I made a last minute call and done the job well.
Not an issue,
by the first day, actually. And I request our principal, sir, Dr. M. Niranjan Babu, sir, to present a token of appreciation in the memento and certificate to Dr. Rastosh, sir. So, just a moment, please, sir.
Yeah. Hello.
Yes, sir.
Yeah, thank you. Thank you, Ashutosh sir. I think nice presentation. I think always we will remember Naipur, Kolkata, because our mentors, Ravi sir is there always to bless us. I think in this group you are also joined with us.
Yes.
Very nice. Convey my regards to sir.
Okay, definitely. I will do that.
Very nice presentation, sir. See you next time and have a good Monday. I think
Right.
whatever the valuable suggestions and whatever the ideas you have given today, definitely it is useful for our pharmacy fraternity. So have a good day, sir. Namaskar, and have this memento for our token of respect.
Yeah. Okay,
So
thank
definitely
you.
we will send this memento to you within a week. Thanks.
Thank you so much. It was a pleasure was
Namaskar.
mine, and you can reach out to me if anytime you need my support.
जरूर सर.
Yeah. Thank you so much.
लोग आप जैसे आदमी से definitely connect करते हैं. So have a nice day, sir.
Yeah. Thank you so much, and pleasure was mine. Thank you.
Yeah.
Thank you, sir. So grateful to you, and happy to connect with Sound Hills College of Pharmacy. And I hope this connection will continue ahead in our future days too, sir.
Definitely, ma'am. Thank you so much, Dr. Suchitra.
Thank you, sir. Namaste.
Okay. Namaste.
Namaste, sir. And we'll be just sending the memento and certificate to you.
Yeah. Fine. Thank you, ma'am.
Thank you so much, sir. Thank you for joining. And faculty members are requested to stay for the third session of this day 1 STTP, and the session is by Dr. Mahendran sir. And the session is going to be scheduled at the time of 2 p.m. So I'm just immensely requesting all the faculty members to be here for the meeting by 2 o'clock exactly. So your attendance will be automatically recorded from the Zoom. So don't worry about your feedbacks and don't worry about your attendance links. So it was automatically recorded and taken up from the Zoom. And those who are attending and following all the requirements of generating the e-certificate can be available to get the certificate. Like 80% of attendance is mandatory, and also the 60% of marks in the end test. So the one who qualifies to these two things can be eligible to avail the e-certificate. So try to understand the norms given by the AICTE and to follow accordingly. So requesting all to be available by 2 p.m. Thank you so much, everyone. Have a good lunch. Bye-bye.